Hi, good morning. Myself, Dr. Mashu. Today, I would like to discuss immunological screening for recombinants. Okay, immunological method used for the selection of recombinants. The next technique is a immunological screening. Okay, next technique is a immunological screening. Okay, in this technique, the desired recombinant clone can be isolated by detecting the protein or a polypeptide synthesized by a gene. Okay, in the previous techniques, we isolated the clones by detecting the sequence present in a recombinant DNA. But here, we'll detect the protein or peptide synthesized by a gene. Okay, that means in this technique, we'll isolate the desired recombinant DNA clone by detecting the protein or a polypeptide synthesized by a gene. Okay, in this technique, in that previous technique that we need that colony hybridization and the plaque hybridization, we need that DNA process. But here, instead of that, we need specific antibodies. Okay, specific antibodies we are required for the immunological screening of recombinant clones. Okay, these antibodies will be highly specific to proteins produced by this gene. Okay, protein produced by genes. Okay, that means this antibodies will be monoclonal and that will be specific to that proteins produced by the our desired gene. Okay, now we can see the steps involved in the immunological. Okay, now we have the transformed colonies. Okay, we having a transformed colonies. Okay, we having a transformed colonies in a plate. Okay, that we'll kept this plate as a master plate. Okay, this transformant containing plate will kept as a master plate, and we'll take this colonies by replica plating. Okay, we undergo for replica plating using that velvet mounted on a wooden block. Using that, we can make a replica plate into nitrocellulose membrane. Okay, we'll get a nitrocellulose exact copy with this colonies copy. We'll get in a nitrocellulose membrane, and we'll lyse this cell. Okay, we'll lyse this cells present on the nitrocellulose membrane. Okay, we'll lyse the colonies cells present on the nitrocellulose membrane, and will allow the proteins present in this colonies on this nitrocellulose membrane. Okay, allow this proteins to bind over this nitrocellulose membrane, and remaining part of the cells will be removed. That DNA or protein, DNA and RNA will be removed, or lipids will be removed. Only this colony containing proteins will be attached on this nitrocellulose membrane. Okay, this proteins colony containing proteins will be attached on this nitrocellulose membrane, and to that, okay, to this nitrocellulose protein containing nitrocellulose membrane will add primary antibody. Okay, we will add a primary antibody into this nitrocellulose membrane. Then that primary antibody will be highly specific to our the protein produced by our desired gene. Okay, protein produced by a desired gene. So this primary antibody will be bind over the this desired protein area. Okay, desired protein area or antibody will be bind. Then we'll add a secondary antibody. Okay, uh, after adding this primary antibody, we'll wash to remove the unbound antibody from this nitrocellulose membrane, and after the washing, we'll add a secondary antibody. Okay, we'll add a secondary antibody, and this secondary antibody will be specific to specific to our specific to primary antibody. Okay, this antibody is specific to our primary antibody, and the secondary antibody will be labeled. Okay, secondary antibody will be labeled. When this secondary antibody will add to this nitrocellulose membrane, this secondary antibody will be bind with the primary antibody is present over here. Okay, this primary antibody present over here. So this secondary antibody then will wash this nitrocellulose membrane. That time this unwound secondary antibody will be removed. Then there will be only bound antibody is there. Secondary antibody is there. So this secondary antibody will bind here only because primary antibodies bind here only. Okay, that's why the secondary antibody bind on this only, and the other remaining antibodies will be secondary antibodies will wash over here because there is no other primary antibody, and we'll wash. Okay, we'll wash and we'll check. Okay, here enzyme is the, and we'll add is this enzyme substrate. Okay, we'll add the enzyme substrate. That time this enzyme will produce the degrade this using that substrate. This enzyme produce color. Okay, this enzyme produce color. That's color indicate the positive colony. That's color indicate the positive colony. You can compare this spot here in the master plate. Here this is a spot. Okay, that means this colony containing the desired our genes protein, desired genes protein. So we can identify that this is the desired desired colony. This contain our transgenic or that or recombinant gene. Okay, this means in this technique, first we have a master plate that's containing a transformants, and this colonies will be transferred into nitrocellulose membrane, and will lyse this cells of this nitrocellulose membrane, and will only this DNA this protein will be bound on this membrane, and into this that membrane will add primary antibody. That primary antibody will be highly specific to our desired uh, genes protein. Okay, how this our desired genes protein, and this primary antibody will be bind with the protein that's desired genes protein present in the nitrocellulose membrane, and the remaining primary antibody will be detached from here, and then we'll add a secondary antibody. Okay, this secondary antibody will be highly specific to the primary antibody. Okay, highly specific to the primary antibody. The primary antibody is only present here because this is the only uh, protein that is specific to this primary antibody. That's why this primary antibody is sorry, the secondary antibody is specific to primary antibody. Then this secondary antibody will be bind on this primary antibody. Okay, because secondary antibody is specific to primary antibody, that's why the secondary antibody will be bind on the primary antibody attached on a specific protein. And this secondary antibody, okay, secondary antibodies are the labeled antibodies. Okay, secondary antibodies are labeled antibodies. When we add substrate into this nitrocellulose membrane, that time this enzyme will convert, use that substrate, that will produce a blue colored, okay, that will produce a, oh sorry, that produce a colored compound. Okay, that will produce a colored compound. On detecting the color formation area, we can isolate which colony. Okay, here you can see, here you can see the primary antibody is attached on into that secondary antibody is also attached here. Here there is no specific antibody, the specific DNA protein is there. That's why there is no attachment of primary antibodies here, and that's why here also there is no secondary antibodies attached here. Only secondary antibody attached here because primary antibody is there, and this enzyme will produce colored compound when added substrate. And here this location we having the colored colony. Okay, will colored area. So this location we can compare with the our mass plate. We can identify which colony having the our desired transformed our desired recombinant DNA is present. Okay, that means in this mechanism we'll isolate the recombinant by depending on the protein synthesized by our desired DNA. Okay, this is the immunological screening of recombinants. I hope you understood this class.
So thank you for listening my class. Thank you.